Assalamu alaikum, this is Zaina doing lipids for foundation here. Inshallah, you benefit from this. Okay, so to begin with, what are lipids? Lipids are substances that are found in living things and they are soluble in non-polar solvent. Simple. So, when we say non-polar solvent, examples of that could be ether, chloroform, or acetone. So, why exactly do we care about lipids? Okay, so lipids are vital energy storage molecules, whether that be in animals or in human beings. So you can think of adipocytes storing fats in fatty tissue. Okay, they're also chemical signals, vitamins and pigments, and they are a part of the membrane structure, and we'll go into that later. So we're going to explore the 6 lipid classes and talk about them each individually. So fatty acids, to start with, are the foundation of all structures of phospholipids and triacylglycerols. And what we mean by that is, if you recall, triacylglycerols are made up of fatty acids and glycerols, so they're just components of that. Okay, and it's important to make the distinction that fatty acids, unlike other macromolecules, cannot form polymers. So for example, when you think of proteins, amino acids form polypeptides, okay? So amino acids to peptides to polypeptides, but in the case of fatty acids, you cannot form polymers. So fatty acids are unbranched hydrocarbon chains, and the length of the hydrocarbon chain is variable, so it's from 12 carbons and above. And of course, with a greater number of carbon atoms, you will have a higher melting point because there's an increased amount of the intermolecular forces, so more energy is required to overcome that. Fatty acids are also monocarboxylic acids. So if you look at the carboxyl functional group that's highlighted in yellow, that is a part of the polar head of the fatty acid structure. So now you might think, if we have a polar head, then why are fatty acids non-polar? That's because the long hydrocarbon chain overcomes that, and the polarity of the head is no longer significant. Okay, so in terms of naming the fatty acid, there are 2 naming systems. We'll start with the numbering system since it is simpler. So the numbering begins from the carboxylate group. The carboxylate group will be assigned carbon number 1 and the adjacent one 2 and so on. Okay, for the Greek system, the carbon that's adjacent to the polar head will be the alpha carbon, and the one next to that is the beta carbon, and then omega carbon is the terminal methyl group on the far left. Okay, so fatty acids are classed into saturated versus unsaturated fatty acids, and then within unsaturated fatty acids, the configuration is either cis or trans. So saturated fatty acids contain only one carbon-carbon single bond. They contain no carbon-carbon double bonds. The entire structure contains carbon-carbon single bonds. For unsaturated fatty acids, you will have one carbon-carbon double bond if it is monounsaturated. If it's polyunsaturated, you will have more than one carbon-carbon double bond, right? And because of unsaturated, because it's a double bond in unsaturated fatty acids, you could have a trans or cis configuration. When the hydrogen atoms are on opposite sides, that is when you have a trans configuration, and when they're on the same side, it is a cis configuration. So if you compare saturated and unsaturated fatty acids, saturated fatty acids are solids, and unsaturated fatty acids are liquids, so they have lower melting points. And that's because unsaturated fatty acids do not pack as closely, okay, because there's a kink in the chain, so less energy is required to overcome intermolecular forces, so there's a lower melting point. Alright, naturally, okay, fatty acids have an even number of carbons in the hydrocarbon chain, and they exist naturally in the cis configuration, so that causes a kink in the fatty acid chain. However, most fats that we consume are trans in the diets, and that's just because of the processing, chemical hydrogenation treatment in food processing. So an example of that is margarine, which is the fat that we regularly consume. Okay. Unsaturated fatty acids are also susceptible to oxidation. So you could see that with oil being rancid, olive oil going bad, or affects, and it's also the oxidation affects the cell membrane. Now to compare fatty acid synthesis in plants and bacteria and humans and animals, it's very simple in plants and bacteria. Synthesis requires no exogenous products. It's just produced endogenously, so it's synthesized from acetyl-CoA. However, in humans and animals, there are two classes that we need to keep in mind. Non-essential fatty acids are synthesized in the body, and they are mostly saturated fatty acids and some monounsaturated. However, with essential fatty acids, you need to acquire them exogenously, so through the diets. And an example of that is omega-6 and omega-3. Why do we need to acquire them exogenously? Because there are no enzymes for their synthesis present in our body. That is not to say that we cannot modify the exogenous fatty acids, because we can still modify them as animals and human beings by adding 2 carbon units and introducing double bonds. So moving on, as you can see here, the table, you'll have to memorize some of the structures and their common names. And if you know the structure, you'll be able to know the abbreviation. And you have to memorize the ones that your doctor indicated. Okay. The abbreviation comes from 2 things. So first of all, N1 is the number of carbons, and N2 is the number of double bonds. And then for the small number on top, alright, you take the total number of the carbon atoms and you subtract the number of the last site of the carbon-carbon double bond. So that is how you establish the number that is next to the triangle on the top. Omegas are actually very important antioxidants in the body. And as I'm sure your doctors have emphasized, palmitoleic acid and oleic acid are the most abundant of the fatty acids in the body. Because omegas are important antioxidants, they prevent beta oxidation. So for the reactions of fatty acids, there are 4 main ones that you need to be aware of. To begin with, dehydration and hydration, because it is a reversible reaction. So it's just essentially the formation of triglycerides. When glycerol and fatty acids react together, that is dehydration, and a side product or a byproduct is water. So as you can see, an ester bond is formed. The second reaction we're interested in is hydrogenation. So for example, plant oils are hydrogenated to trans fatty acids, and this is a side product of margarine production, which we mentioned earlier. For the third reaction, protein acylation, fatty acids are covalently bonded to acylated proteins, and this facilitates interactions between membrane proteins and their hydrophobic environment. The most common protein acylation reactions are myricylation and palmitoylation. Okay, and they influence a variety of structural and functional properties of proteins. And lastly, oxidation most commonly occurs with unsaturated fatty acids. So now on to our next lipid group, eicosanoids. Okay, they are short-lived signaling molecules, so that's why we say they are similar to hormones. Okay, they are produced in mammalian tissues. However, they are produced in small amounts. Okay, and they have in general an autocrine effect. So if you can recall from biology, an autocrine effect is they produce it themselves to affect themselves, to act on their own cells. Okay, and sometimes paracrine, so they travel short distances to affect nearby cells. Omega-6 and omega-3 are the main precursors of eicosanoids. So eicosanoids are produced after the omega-6 and 3 are released from the membrane phospholipids by an enzyme called phospholipase A2, which we will look into phospholipases soon. Alright? Now, eicosanoids are actually implicated in diseases like myocardial infarctions and rheumatoid arthritis. The difference between omega-6 and omega-3 is really important, and it's crucial to understand the ratio and why we care about the ratio. So omega-6 is actually pro-inflammatory, so it's sort of بتحفز, it's stimulating inflammation. Omega-3 is anti-inflammatory, okay? So usually 1:1 or 1:4 is a healthy W6 to omega-6 to omega-3 ratio. However, when this ratio increases, for example in developed countries because of certain diets, it's unfavorable and inflammatory reactions occur more often in the body, and that will increase the likelihood of chronic diseases. And the ratios in the diet will influence the amount of inflammatory and anti-inflammatory eicosanoids synthesized, which is why this is so important. So like we've said, eicosanoids are derived from omega-6 and omega-3, but how precisely? Omega-6 and omega-3 will form some precursors to the eicosanoids themselves. So omega-6 linolenic acid will form arachidonic acid via elongation and desaturation reactions, and this is what mainly forms our eicosanoids. And omega-3 or alpha-linolenic acid, linoleic acid, will form DHA, which is what forms our eicosanoids. Now the effects of omega-3 is a decrease in the triacylglycerols in the blood, a cardiovascular health boost, decrease in blood pressure, and decrease in platelet aggregations, which are all excellent and positive effects on the body. So in general, eicosanoids are mediators of smooth muscle contraction, inflammation, blood flow regulation, and a lot of positive functions. So how do we name eicosanoids? The first two letters are classing the eicosanoid type, and it's most commonly PG, TX, and LG. The third letter is the type of modification made to the parent compound of the eicosanoid. So A indicates the addition of a hydroxyl group in an ether ring, and B indicates two hydroxyl groups. And finally, we name eicosanoids by the number of double bonds. So eicosanoids are of three types, most commonly. They are prostaglandins, thromboxanes, and leukotrienes. So you need to know a few features of the structures of these eicosanoids and their roles in the body. You might be familiar with thromboxanes from biology. So thromboxanes are involved in platelet aggregation and vasoconstriction, and they are generally synthesized by lymphocytes. Now, the key structural feature in thromboxanes is the cyclic ether, which you can see. For leukotrienes, they are chemoattractants, so they attract white blood cells from the immune system. They're vasoconstrictors and bronchoconstrictors, so two constrictive functions, which is like thromboxanes being vasoconstrictors as well, and edema. So for leukotrienes, there's a triene group. For prostaglandins, they are involved in inflammation, digestion, and reproduction, and they have a signature cyclopentane ring, which you can see on the left of the structure, as well as hydroxyl groups at C11 and C15. So moving on now, for triacylglycerols, they are either fats or oils, and this just depends on the state of the hydrocarbon chains. So if there were a higher proportion of saturated fatty acid hydrocarbon chains, then they are fats. If there are higher proportions of unsaturated fatty acid chains, then they are oils. And this goes back to the melting point thing that we discussed with intermolecular forces and kinks in chains earlier on. So triglycerides are actually neutral fats. They are hydrophobic and they have no charge. Their function is energy storage, insulation, water repellent, so they're found in the fur of animals on their skin, and so on. So why exactly are they good for energy storage? And why are they better than glycogen? Because fatty acids are hydrophobic, they coalesce. So what does coalesce mean? If you can see these circles on the top right and the left, the lipid droplets actually come together and they aggregate. Just think of the oil and water experiment, okay? They coalesce into anhydrous droplets. And because triacylglycerols are more reduced, they release more electrons when oxidized, and that's what makes them higher energy storage molecules than glycogen. In terms of the structure of triacylglycerols, it's really simple. Triacylglycerols or triglycerides are formed by the reaction of glycerol and free fatty acids. Okay, and this is essentially an esterification reaction because ester bonds are formed and water is released as a byproduct. So the fatty acids can be saturated and unsaturated, like we discussed, which makes them fats or oils. We have monoacylglycerols and diacylglycerols, which are metabolic intermediates. Monoacylglycerols are when you have one fatty acid and a glycerol, and diacylglycerols are when you have two fatty acids and glycerol. So what are waxes, and what distinguishes them? Waxes are actually esters of long-chain fatty acids and long-chain alcohols. So they're literally the same as triacylglycerols, but the distinction is the fact that the chains are very long. And for examples of waxes, you can think of waxes on the fur and skin of animals, waxes in honey wax, right? Like in beehives, and waxes that coat fruits naturally. Next lipid group is phospholipids, and this is something I'm sure you're familiar with. So phospholipids are actually amphipathic molecules. Okay, what does amphipathic mean? So it means they have dual properties. They have a polar head, which is highlighted here, and it's made of glycerol and a phosphate group. So the charged phosphate group will contribute to the polarity. And they have non-polar tails, which is made up of two fatty acids. Okay, that is the structure of a phospholipid. So phospholipids are involved in membrane formation. If you think of your phospholipid bilayer, they're also involved in emulsification and they're surfactants, so they act as detergents. Phospholipids can actually spontaneously rearrange, depending on the environment that surrounds them, into micelles or bilayers. So if you look at the circular structure on the left, that is a single layer, a unilayer. It is a micelle. If you look on the right, that is a bilayer. So phospholipids are of two types. They can be phosphoglycerides or sphingolipids. The difference between that and a sphingolipid is a sphingolipid has sphingosine instead of glycerol. Okay, so sphingosine instead of glycerol. So phosphoglycerides is when glycerol can bind to three fatty acids or two fatty acids and a phosphate. And the simplest phosphoglyceride is phosphatidic acid. And phosphatidic acid has derivatives. So the derivatives come about when different groups are esterified to the phosphate. Sphingolipids are when sphingosine can bind to one fatty acid, which makes it a ceramide. And when a ceramide has a phosphate, it's called a sphingomyelin. So let's look into phospholipases that we alluded to earlier on. So phospholipases are actually enzymes. They hydrolyze ester bonds in phosphoglyceride molecules, and they're involved in membrane remodeling, so they change the ratios of phospholipids in the membrane to enhance or affect fluidity. They're also involved in signal transduction initiation. So this enzyme's activity is the beginning step of signal transduction. They're involved in digestion, so specifically pancreatic phospholipases exist in the small intestine. And they're also involved in damaging membranes. This is a negative effect that isn't actually initiated by our body itself. It's from bacterial alpha toxin or snake and bee venom. Okay. And we have two phospholipases, phospholipase 1 and phospholipase 2. They hydrolyze the ester bonds, as you can see in the circle. And the products of phospholipase 1 and 2 are fatty acids and lysophosphatide. A lysophosphatide is just a phosphoglyceride which is lacking a fatty acid. So phospholipid A and phospholipid 1 and phospholipid 2 have a similar function, it's just they target different ester bonds. Okay. We also have phosphodiesterases, which are PLC and PLD, and they yield diacylglycerol and phosphatidic acid, respectively. So PLC yields diacylglycerol and PLD yields phosphatidic acid. So because we looked at phospholipids, now we will look at sphingolipids and glycolipids. So sphingolipids are sphingosines and ceramides. And as you can see on the top right, the structure of the sphingosine and the ceramide. So sphingosine is actually a long chain amino alcohol, and our ceramides are also precursors of glycolipids. So where carbs are attached to ceramide through the glycosidic bond is a glycolipid. So when a carb is attached to a ceramide via a glycosidic bond, glycolipids come about. And an important point that doctors tend to emphasize is the lysosomal storage disease involved with sphingolipid metabolism, Tay-Sachs disease. Okay, so what we have is a deficiency in the enzyme beta-hexosaminidase A, and this enzyme is no longer able to metabolize ganglioside, which is a sphingolipid, and the cells are going to swell and die. And symptoms of that are things such as blindness, muscle weakness, seizures, and mental retardation. So glycolipids are carbohydrates and ceramides, as I've already mentioned, and the classes are cerebrosides, sulfatides, and gangliosides. So moving on to isoprenoids. Isoprenoids consists of repeating 5-carbon isoprene units. And under isoprenoids, we have terpenes and steroids. So terpenes are classified by the number of isoprene units. You could have monoterpenes and you could have tetraterpenes. Monoterpenes is when you have 2 isoprene units, and an example of that is perfumes, and tetraterpenes is when you have 8 isoprene units, and an example of that is pigments. So carotenoids. So if you remember earlier on, we mentioned the function of lipids is vitamins and pigments. So carotenoids are an example of those pigments, and they are tetraterpenes under isoprenoids. Okay, fat-soluble vitamins and steroids. So vitamins A, D, E, and K are lipid-soluble, and they originate from isoprenoid pathways. Mixed terpenoids are vitamin K and E. They have a non-terpene group as well as an isoprenoid group. So finally, for the last lipid group, we have steroids. Steroids are derivatives of triterpenes, and they consist of 4 fused rings, and that's a signature feature of steroid molecules. And it's also called the steroid nucleus because of how standard it is. Okay, so cholesterol is the most important steroid, and it's classed as a sterol because the C3 is oxidized to a OH. Cholesterol is commonly found in cell membranes. We know about this. It affects the membrane rigidity, and it's a precursor for steroid hormones, vitamin D, and bile salts. So cardiac glycosides are carbohydrate esters of steroids, and you have some examples here. They're known for their ability to increase the force of cardiac muscle contractions. So we can see a proper application of steroids in the cell membrane and from a cardiac aspect. The mechanism of cardiac glycosides are the inhibition of sodium-potassium ATPase membranes. And this will reinforce the effect of increasing the force of cardiac muscle contraction. Finally, let's look at some cholesterol derivatives. So these are hormones you will become so familiar with later on in your studies. So it's important to sort of understand how they come about as derivatives and what their function and application is. So progesterone is a female hormone that's predominantly found in females. Testosterone is a hormone that's predominantly found in males. Estradiol is basically estrogen, which is a predominant hormone in females. So these 3 hormones are primary and secondary sex hormones that are involved in primary and secondary sexual characteristics and reproductive behaviors. Okay. You also have aldosterone, which is a mineralocorticoid hormone. It's produced in the adrenal cortex, and it's involved in the regulation of plasma concentration of ions, such as in sodium, potassium. Okay. Cortisol is a metabolic hormone, and it's a glucocorticoid, and this is also produced by the adrenal cortex. It regulates fat, carb, and protein metabolism. Finally, cholic acid, which is a bile acid, is a part of bile salts in the liver. It aids in fat digestion and vitamin absorption in the intestine. That is all for lipids. Thank you so much for listening.
Hello everyone. Today I'll be talking about lipoproteins. So first of all, what is lipoprotein? Lipoprotein is a structure that that has a function of transporting lipids in the blood from an organ to another. Okay. When we say this lipoprotein should transport lipid inside in the blood, we know the fact that a lipid is a hydrophobic, something that is hydrophobic. So how will we be able to transport it in a blood which is similar to water, or it has water in its components? Okay, that's where lipoproteins come into function. Okay. what is, in order to know how does the lipoprotein transport this lipid? First of all, we need to know the structure of the lipoprotein. It consists of what exactly? Okay. So as we know, the blood consists of a plasma, as we said before, which is good. And the lipoprotein is transporting lipids which are hydrophobic, thus they are not soluble in the blood, hence they cannot be transported. So how the lipoproteins manage to transport those lipids? Here is the question. So let's try to answer it by knowing the structure of the lipoprotein. Okay. Lipoprotein consists of an outer layer and an inner core. Inner core means yani, something central. Imagine the lipoprotein as a ball. Okay. The center of it is the lipid, and the outer layer of it is a protein. Okay. So the inner core is a triglyceride. It could be triglyceride, it could be cholesterol, it could be cholesterol esters, any lipid. The outer layer consists of an phospholipid monolayer. What do we know about any cell membrane? The cell membrane is made up of phospholipid bilayer, 2 layers of phospholipid, and not a monolayer. So that's the difference between lipoprotein, phospholipid, and the phospholipid of the cell membrane. Okay. As we said also, the outer layer consists of a protein. So the outer layer is a phospholipid and a protein. The inner layer is fats, lipids. Okay, what are those proteins in the outer layer? They are the apoproteins. Okay. هلا مثل ما بقول المثل صورة تغني عن 1000 كلمة. Let's imagine the structure. Okay. So this thing in purple is the lipids, triglycerides, cholesterol, name it and okay. The outer layer is a phospholipid monolayer and not a bilayer. This structure in these things in yellow, they are the phospholipids. Okay, what is this blue structure? It is the protein, the apoprotein we talked about earlier. Okay, how does this structure how does it help us answer the previous question? You know, how do we transport this lipid? The outer layer is a lipid, so basically the lipid is soluble. No, I mean the outer layer is a protein and phospholipids. They are soluble in the blood. Okay. What about the inner? It's the fat. So basically what happens is that this fat is insulated completely from the blood. So it does not get into contact with the blood at all. So the outer structure is soluble and it will be transported. However, the inner is insoluble, but it's insulated from the blood. So it does not contact the blood at all, and thus it can also be transported. Okay. So that's about the structure of the lipoprotein. Okay. We talked about in the previous image, as we said, this blue structure is the protein, the apoprotein. Well, we have subtypes of apoproteins. We have B, C, and E, and there are many other lipoproteins. But our focus is in about B, C, and E. Okay? B, we could have B48, B100. It differs depending on the type of lipoprotein. Lipoproteins, we have chylomicron, we have VLDL, we have LDL, and we have HDL. I'll give you a rule from the beginning. HDL does not have ApoB at all in its structure. We are left with VLDL and chylomicron. Which is which? VLDL has B100 On the other hand, chylomicrons have B48. Okay. طيب بعدين أبو عندنا C. There are many other subtypes of C, but our main focus is about C2. This is the most important ApoC, apoprotein C. طيب. And we have E. It's only E. There is no subtypes of it. طيب. You can see here, it's mentioned that C2 activates lipoprotein lipase in blood vessels endothelium. طبعا, till now you won't understand this sentence. So just give it a chance. Then at the end of the lecture, we will come back to this and we will re-explain it. And E helps in the absorption of chylomicron remnants by the liver. Also, you won't understand this sentence right now. Then we will come back to it. Okay? So now we talked about the apoproteins, which are these blue structures. We talked about them. Now we need to talk, and we talked about the triglycerides and cholesterol. The phospholipid, we said it's a monolayer. However, now we need to talk about each and every specific lipoprotein. We need to talk about every lipoprotein. Chylomicron in specific, VLDL in specific, HDL in specific. Okay? طيب. هلا, as you can see, chylomicron, CM means chylomicron. This is VLDL. Ignore IDL, ignore it. We have LDL and HDL. طيب. هلا, this arrow shows us that as we move downward from chylomicron till HDL, the density increases. طيب, هلا احنا, what do we know about the density? The density is determined by the protein content in the lipoprotein. طيب, what do I mean by this? I'll come back to this. This protein, which is the apoprotein, whenever the lipoprotein has lots of amount of this, like now you have only 3 right here, 1, 2, and 3. If you have like 16 of this protein, the density of this lipoprotein increases. So we increase the protein content, we increase the density. تمام? Okay. Okay. طيب, تمام. This is the density. C chylomicron is less than 0.95. VLDL is a bit higher. LDL is a bit higher. And the highest density is HDL. HDL has the highest protein content. On the other hand, chylomicron has the lowest protein content. طيب, تمام. One more thing we need to know is that as the protein content decreases, we need to compare now. We said as the protein content decreases, the density decreases. That's right? Yes. Okay. برضو, as the protein content and density decreases, relative to the protein content, the lipids increases. So basically, for example, you have a population of 100 person. طيب, تمام. The 100 people, for example, 50 are lipid, 50 are protein. So they are equal. طيب. Now we increase the protein content. So they became 60 protein and they became 40 lipids. So now the lipid is decreased. طيب. What determines the size of the lipoprotein is the lipid. So what determines the density is the protein content. What determines the size, it's the lipid. So as a general rule, when we move from chylomicron to HDL, the density increases. As we said, when the density increases, the protein content increases relative to the lipid content. So the lipid content is now decreased. And we said the lipid content determines the size. So the size decreases. So the density increases, the size decreases as we move downward. We move downward, density increases, size decreases. Tamam. Now let's talk about each and every lipoprotein. Chylomicron and VLDL are the main carriers of triglyceride lipids. So basically, you go check on VLDL or a chylomicron, move to the, you see here a lot of triglycerides and lesser cholesterol content. Okay, that's the first rule. Type. HDL and LDL are the main carriers of cholesterol and cholesterol esters. Like the previous image, instead of triglycerides, we will see a lot of cholesterol. We will find a relative amount of triglycerides, but they mainly transport cholesterol and cholesterol esters. Okay? And also, one more thing. Chylomicron has this specific function, which is transporting exogenous lipids. Type, what do I mean by exogenous lipids? Lipids in our body, either the liver synthesizes the liver, either the liver synthesizes the lipid, or we take the lipid from outside the body. We take it like in the food. Okay. So endogenous is any lipid produced by the liver, synthesized by the liver without eating something. It's by itself. The liver synthesizes. Okay. On the other hand, exogenous, anything you eat, that any fat, meal, lipid, that's considered as exogenous. Okay. So chylomicrons are responsible for transporting exogenous lipids. Lipids that are not synthesized in the body. Lipids, we take them in the meal. Okay. Type, we eat a meal. Where the food goes, esophagus, down the GI tract, to the stomach, and then to the small intestine. In the small intestine, it will get absorbed. طيب. We need to know this thing. Chylomicrons are synthesized in the small intestine. Okay? So basically, the food comes to the chylomicron. So the chylomicron lives in the small intestine, the food comes to the chylomicron. And then what will the chylomicron do? It will inhale or it will inject, it will eat all the food. That's what I'm saying. Okay. Ignore that. Inhale well. Okay. So any exogenous lipids in the meal, they go to the small intestine where they meet with the chylomicron. On the other hand, VLDL transports endogenous lipids. طيب. What are the endogenous lipids? Endogenous lipids, as I said, they are the lipids that are synthesized by the liver in our body. Tamam? So they are synthesized in the liver. VLDL transport endogenous lipids that are synthesized in the liver, and also VLDL itself, this lipoprotein called VLDL, is synthesized by the liver. So that we should know. Chylomicron are synthesized in the small intestine, VLDL is synthesized in the liver or by the liver. Okay. طيب, let's start by chylomicron. I'll tell you the story of the chylomicron. And as we said, they are synthesized by intestinal mucosal cell. We shared that before. Similar to other lipoproteins, they consist of an outer layer. We mentioned that. Outer layer consisting of phospholipid monolayer and the specific apoproteins, the blue structures in the first image. And we said also previously, I want to go back You remember what I said here about B, which is the apoprotein? We said if it's a B100, it's VLDL. It's in the VLDL. B48 is in the chylomicron. So now we know that apoprotein B48 is in the chylomicron. Now we know this apoprotein, but is it the only apoprotein in the chylomicron? No, we have others, but we still didn't mention them. Okay. Tamam. We said at the beginning, chylomicron is released by small intestine. We mentioned that as immature. What do I mean by immature? Immature يعني it does not have a function till now. It's a nascent, we call it. Nascent chylomicron. And it has only apoprotein B48. We mentioned that a few seconds ago as an apoprotein. طيب when the chylomicron has only apoprotein B48, it's immature. When do we call it mature? Chylomicron is said only to be mature when HDL gives the chylomicron ApoC2 and ApoE. طبعا till now you don't know what I'm talking about because we still didn't talk about the story of chylomicron. يعني من الآخر chylomicron is immature if it has only apoprotein B48. When it becomes mature, when she gets ApoC2 and ApoE. It's given by the HDL. So when we call it a mature, when chylomicron has B48, C2 and E. That's where we call it a mature chylomicron. طيب. And as we said earlier, they transport exogenous lipids from amine. طيب تمام. Now we'll talk about the real story, the real, okay. You have here food. Food goes down the gastrointestinal tract to the stomach, to the small intestine. Here in the small intestine we have the small intestine cells. They are responsible for absorbing food and okay, nutrients and everything. طيب. Here is the food we took. It will get absorbed where it meets chylomicron. Chylomicron, here it is. And as we said, it's immature, where it has only apoprotein B48. طيب, what will the chylomicron do? It will uptake this food, and طبعا the food will be broken down into triglycerides. As we said, برضو, chylomicrons mainly are carriers of triglycerides. So the food is broken down into triglycerides. What is a triglyceride? Here's the structure of it. Okay, and we have 3 fatty acid chains. Okay, this thing will be uptaken by the chylomicron, the immature one. It will join it. Tamam? So that now we have the chylomicron with the apo B48, which means that the chylomicron is still immature, and it uptakes the triglyceride into it. So the chylomicron has the triglyceride inside it, and the apo B48, which means it's still immature. طيب تمام. هلا the chylomicron then gets absorbed into the lymphatics, الجهاز الليمفاوي بما معناه. طيب, after it gets absorbed to the lymphatics, it goes to the blood, the circulation. تمام. خلاص, it enters the blood. Okay. Now when it enters the blood, we will continue the story in the next slide. Okay. Ignore all the drawings. Just focus. We are still here in the blood. This blue structure is the chylomicron. This thing. Okay. Tamam. With the apo B48, which means it's still immature. Okay. طيب, the chylomicron, as soon as it enters the blood, it meets something called HDL, which is a lipoprotein. طيب, what will HDL do? it will convert the immature chylomicron into a mature chylomicron. How will it do it? It will do it by giving it the apoproteins C2 and E. So basically, we have the chylomicron with the apoprotein B48 at the beginning. It meets the HDL. The HDL gives the chylomicron ApoC2 and ApoE. So now, what will happen? The resultant is a mature chylomicron. So the mature chylomicron has it's originally apoprotein B48. It has also the apoprotein C2 and it has ApoE. See? Apo B48, C2 and E. Now it's a mature chylomicron. طب what will this chylomicron do? At the beginning, in the small intestine, we said it uptake the triglycerides into it. طب هلا what do we expect it to do? It will transport these triglycerides to the adipose tissues and to other tissues in the body. تمام? That's its function. Here's the mature chylomicron with the B48, E and C2. تمام؟ طيب. هلا we need to know what is the function of this apoprotein C2. Why the HDL gave the chylomicron this apoprotein C2? What will it do? Okay. Apoprotein C2 activates. طبعا here we reach طبعا the tissues. Here's the adipose tissue. طيب. Now what do we expect from the chylomicron to do? It will release the triglycerides into the adipose tissue. طب we need something in order to do so. We need something that extracts and breaks down the triglycerides into 3 fatty acids. We need to know this general rule. Okay, the triglyceride in the chylomicron cannot pass through the endothelium of the blood vessel. It cannot do so. It has to be broken because it's a big structure. How will we transport it in this small endothelium openings of the blood vessel? We cannot do so. We need to break down this triglyceride in the chylomicron into 3 fatty acids which are small structures. تمام? What will do this job? Something called lipoprotein lipase. Okay, I'll go to the next slide in order to explain it. تمام. Here is the mature chylomicron. Okay. Here is the triglyceride in the chylomicron. The chylomicron will release these triglycerides in order for them to pass down to the adipocytes. Okay? Tamam. We said they are a bulky structure, very big structure. They cannot pass like this through the blood vessel wall. What will we do? We will try to break the triglyceride into 3 fatty acids. So what structure will do this? Something called LPL, lipoprotein lipase. طيب. It will break down this triglyceride. This is the first fatty acid, second and third fatty acid. But there is something we forgot about. Lipoprotein lipase will never be active except if there is an apoprotein that activates it. Okay, I'll go back to the previous slide. Here we go back to this LPL, lipoprotein lipase, which is responsible for breaking down the triglyceride into 3 fatty acids. طيب, which apoprotein activates this LPL? It's apoprotein C2. Apoprotein C2 is the one responsible for activating the LPL in order for the LPL to break down the triglycerides into 3 fatty acids. In order for the 3 fatty acids to pass through the blood vessel wall into the tissues. Without this C2, the triglyceride will stay in the blood and it will cause many problems. So after ApoC2 activated after the ApoC2 activated the lipoprotein lipase, it became an active lipoprotein lipase, active LPL. This active LPL will break down the triglyceride into 3 fatty acid chains, cuts into 3 fatty acid chains. Then they can enter into the adipocytes through the blood vessel wall, because now they became smaller structures, not a one bulky structure. Okay. طيب تمام. خلاص, we are happy. We gave the blood vessel. خلاص, we donated our triglyceride. خلاص, we don't want it. We gave it to the tissues. Now we are free of triglyceride, or not free, but we have less amount of triglyceride in our content. So basically, the chylomicron now, after it released the triglyceride, it has less or without triglycerides. تمام? Now what will the chylomicron do? خلاص, I finished my job. What will I do? The chylomicron will go back to the liver. طيب. It will go back to the liver. What can help the liver to reuptake this chylomicron? طبعا, we call this chylomicron, after it released the triglyceride, chylomicron remnant. What does remnant mean? Remnant from the word remains. What remains of the chylomicron? خلاص, we release the triglyceride, and that's what's remaining of the chylomicron. We need to reuptake it by the liver. The apoprotein E is responsible for the liver in order to recognize this chylomicron remnant. What do I mean by this? Okay. Apoprotein E, without it, the liver won't uptake this chylomicron. So we need this ApoE in order to tell the liver, I belong to you, take me. Okay? That's how to explain it. Like, the ApoE makes the liver recognize the chylomicron, so the liver takes the chylomicron in. Without the ApoE, the liver won't take the chylomicron inside it. Okay. So now, when I go back to the previous slide, I said you won't understand it. Now I think it's very clear. Apoprotein C2 activates lipoprotein lipase in the blood vessel endothelium. خلاص, it's obvious now. And ApoE helps the absorption of chylomicron by the liver. That's it. Uptake of chylomicron by the liver. تمام. خلاص, that's the end of the chylomicron story. That's it for the chylomicron. طيب, VLDL has the exact same story of chylomicron. There is no difference. VLDL instead, it's produced in the liver, as a contrary of the chylomicron which was produced in the small intestine. طبعا, at the liver, it's at the beginning, the VLDL is released by the liver as immature. Similar, exact same thing to chylomicron, immature. And it has only apoprotein B100. What's the difference between VLDL and chylomicron? VLDL, apo B100, chylomicron B48. تمام? And it transports triglycerides mainly, as we said in the beginning. Chylomicron, VLDL, mainly triglycerides, synthesized by the liver to the adipose tissue for storage. Exact same function of chylomicron. Okay, chylomicron, I meant VLDL, is only said to be mature when HDL and VLDL, HDL gives VLDL ApoC2 and ApoE, similar to chylomicron. HDL gave chylomicron ApoC2 and ApoE, now it became active. تمام؟ Okay. بعدين نفس الستوري. After releasing the triglyceride into the adipose tissue, VLDL converts into LDL. Okay, now we need to talk about this in more depth. Converts into LDL. هلا, there's something we should know. Chylomicron, after it released the triglycerides into the tissues, it became a chylomicron remnant. What about VLDL? when it releases the triglyceride, it will be called LDL. Okay, what is this LDL? هلا LDL هسا حكينا VLDL خلاص, we release the triglycerides. What's left inside the VLDL is or what's left is a little amount of cholesterol and proteins. And as we said at the beginning, LDL and HDL have very high cholesterol content, and they are the main carriers of cholesterol. I'll go back to the previous slide. Okay, here we said chylomicron VLDL are the main carriers of triglycerides. HDL, LDL, main carriers of cholesterol and cholesterol esters. تمام؟ now we are left with LDL. We are left with LDL, which has a high cholesterol content. Okay, and very low triglyceride content. And due to this high cholesterol content, it can lead to cholesterol deposition in the subendothelium of blood vessel, which is called atherosclerosis. Tamam. I'll explain this. I'll try to draw as much as possible. Okay. We have, this is the liver. Imagine it as the liver. Okay. The liver حكينا liver produced VLDL. تمام. VLDL now go with what do we have? B100, and we said that was the main difference between it and the chylomicron. تمام. طيب, now the VLDL enters the blood directly. Why directly? هلا الchylomicron from the small intestine, it will go first into the lymphatics and then into the blood. The key difference between it and the chylomicron is that the VLDL goes directly to the blood. Here's the blood vessel. Okay. Okay. And for example, I'll draw the tissues. Adipose tissues. Adipose tissue. تمام. طيب. ال VLDL enter with the ApoB100. Now it meets what? You know what does it mean? The blue guy, the HDL. The HDL, what will it do? It will give it C2 and E. It will give those to the VLDL. تمام؟ طيب. Okay. So now it became mature. VLDL. طيب. Mature VLDL. We reach the tissue. For example, here's the tissue, and what do we have here? What is called lipoprotein LPL. Lipoprotein lipase. We are repeating the same story. What activates the lipoprotein lipase? It's the C2. C2 activates the LPL. خلاص, LPL now became active. Lipoprotein lipase. What will it do? It will break down the triglycerides. In order, here's the triglyceride. I'll write TG. TG means triglycerides. In order for the triglyceride to pass into the adipose tissue as free fatty acids. as free fatty acids. اوكي خلص. mature VLDL now حكينا it has ApoC2, it activated lipoprotein lipase. Okay, lipoprotein lipase, what will it do? It will break down the triglycerides into 3 fatty acid chains. Why? Because the triglyceride was a bulky, something bulky, big, it cannot pass through the blood vessel wall. On the other hand, fatty acids are small and they can pass through the blood vessel wall into the adipose tissue. طيب, what will happen? Now the VLDL, after it released the triglyceride, we are left with LDL. Now it changed its name into something called LDL. LDL is rich in cholesterol. تمام؟ طيب. When it's rich in cholesterol, it can lead to something very bad. What is it? It can make this is the cholesterol, it can deposit it into the blood vessel wall. Consider it as someone who is careless, he's throwing everything on the floor, he doesn't clean, okay. He makes a mess everywhere. What cleans this mess? The HDL. The same HDL that gave the VLDL ApoC2. The same one, this one, HDL. HDL will collect this cholesterol. طيب. HDL will take this cholesterol. Now we have it here. HDL with the cholesterol. طيب. طيب. We have the HDL here. It collected the subendothelial cholesterol. طيب. As it collects it. طيب. هلا HDL in general, any lipoprotein, its main function is to transport lipids in huge amounts. Collect all the lipids and transport it. We need يعني any lipid it finds, it collects. So consider the lipoprotein as a box. It has a limitations. It has a space, and above this space it cannot hold more than that amount of lipids. Okay? طيب تمام. هلا when HDL collects the cholesterol, this cholesterol has a structure which is very bad. Okay. Okay, here's the structure of a cholesterol. It has a OH group on carbon number 3. What? This OH group is very bad. Why? OH is hydrophilic. It attracts water. تمام? طيب. Consider this box as HDL or any lipoprotein. We have this pen. Okay, here's the pen. This is the cholesterol. It has the OH group, which is, we have this cholesterol. It's this pen. It has the OH group. I'll just go back to the slide. The OH group has the, okay. OH group attracts all the water. Okay. I place this cholesterol inside the box. Now, the box has the cholesterol, which has the OH group, attracting all the water inside the box, into the box from outside. Okay? Now the box gets filled with water. طيب. When the box gets filled with water, now the space is completely occupied by water, and just one cholesterol molecule, which is the pen. Is that what we want? No. The purpose of lipoproteins is just to transport lipids, not water. Okay, so basically what we want is to prevent this attraction of water. We want to prevent it. How can we do so? By removing the OH group. طيب. How can we do so? By converting the OH group into a fatty acid chain. The fatty acid chain makes it hydrophobic, makes the cholesterol hydrophobic. It doesn't love water. طيب تمام. فناw the cholesterol no longer attracts water, and we place the cholesterol inside the box, it won't attract water. Now the box can hold a huge amount of lipids and very very little amounts of water, or no water أصلا at all. تمام. We have the OH group. Now it became a fatty acid. it makes the compound hydrophobic, not attracting water at all. Now we can, the box, which is the HDL, can hold lots of amounts of lipids inside it. طيب. هلا there is something إنه حكينا HDL collects free cholesterol in the blood vessel and transport to either the liver for storage or to circulating VLDL or chylomicron. Okay, what am I saying is we have the HDL. It collects the cholesterol from the blood vessel. This cholesterol came from where? From the LDL. LDL depositing cholesterol on the wall of the blood vessel. تمام؟ طيب. HDL came to clean up this mess and taking up this cholesterol inside it. Now, what will the HDL do to this cholesterol? It will either give it to the liver or to a circulating chylomicron, circulating chylomicron and VLDL. تمام. هلا what was let me go back to the previous slide. What was responsible for converting the cholesterol to a cholesterol ester, having the new fatty acid chain added to the OH group on carbon number 3? It was an enzyme called CAT. ACAT, LCAT, whatever. It's an enzyme called CAT. تمام? Okay. Okay, here's our enzyme CAT converting cholesterol into a cholesterol ester. Type CAT can be subdivided into two types, either LCAT or ACAT. LCAT is found in the blood, in anything circulating in the blood, either the blood itself or in the lipoproteins that are moving in the blood. So any lipoprotein, I tell you, VLDL, HDL, chylomicron, they all have LCAT. تمام؟ طيب. On the other hand, ACAT is found in tissues, such as the liver, for example. طيب, why is that important? I go back to this previous slide. I tell you that HDL will give this free cholesterol either to the liver or to a circulating VLDL and chylomicron. تمام؟ طيب. If I tell you that it gives it to the liver, the liver will convert the cholesterol to a cholesterol ester by which enzyme? By ACAT. طيب. If I told you that the HDL gave it to a chylomicron, the chylomicron will convert to this cholesterol to cholesterol ester by which enzyme? LCAT. Okay? Okay, now I'll just do a very, very quick recap. طيب. We have first of all, we have nascent. Chylomicron could be nascent, mature. At the beginning, it's nascent or immature. It has only ApoB48. On the other hand, VLDL has only ApoB100 as immature. طيب. Chylomicron becomes mature. We add to it ApoE and ApoC2. VLDL, same thing. We add to it ApoE and ApoC2. Does HDL have ApoB? No, it does not have ApoB. HDL does not have any ApoB. It has ApoC2 and ApoE. Okay. That's it, and thank you very much.